And welcome to another Wednesday webinar. I missed last week because of the Wise Traditions, Nourishing Traditions Weston Price Conference in Kansas City, which was, I thought, a great event. Lots of people, lots of new people. I got to meet and reconnect with many of you. I appreciate everybody coming up and saying hello to me. So yeah, that was a great event. I hope some of you were able to make it. And I think that's it for announcements, as far as I know. Yeah, so let's move on. So the bulk of today is going to be, as I said, Harold Hillman's 47 questions. And I just ran this by somebody, and the comment was, that's going to be pretty tedious and boring. So it's possible that's true. So why am I doing it? Not to be tedious and boring particularly, but I think, first of all, I think it's interesting. So that's reason enough for me to do it. Second of all, I think it's important not to get this, the work of Hillman out as much as I can. And I've never seen this list of 47 questions, and I don't know that I need to read them all and go through them all. And a lot of them I don't actually know very much about or even what he's really talking about, so I'll probably skip over those. But I think at least one time somewhere, on the web, there should be a description of these 47 questions. This is sort of the compilation of Hillman's work over his career. And I think you'll be able to appreciate, even if you don't understand all the details, like I don't certainly understand all the details myself, but you'll hopefully get a flavor of just how profound his criticism of modern biology and therefore the underpinnings of modern medicine is. And I think that that is actually huge for us going forward, because so much of the activities and the strategies, that's a better word, of modern medicine come directly out of the so-called fundamental facts in biology. And as you'll see, and as I've been talking about for a few years, these fundamental ideas are actually far from fact. And actually the whole field of modern cell biology needs to be reevaluated. And therefore, the whole philosophical, so-called scientific underpins of modern medicine, as you've heard, needs to be reevaluated. So that's why I'm doing it. But because this is potentially kind of tedious and not so much fun, I thought I would start with a clip that's hopefully a little more fun and actually gets at to another principle, which I've been alluding to, but you'll see it in a sort of kind of goofy, funny way. And that is the principle of, does your mind, or maybe your consciousness, or maybe you could say the way you think, or the way you conceive of the world, affect what happens to you? And does it actually affect what you do? And just what happens to you? And so this is a pretty interesting demonstration of just that principle. And I want to just say up front that I actually have no idea whether this is faked or not. It doesn't look like it's fake to me, but I haven't investigated either this guy or this particular video in any great depth. So it could be that I'm about to be faked, and so will you. But if we just take for a minute that it doesn't appear faked, and somebody maybe will look into it, I think it's well worth taking a look at this little clip. If for nothing else, it's kind of fun. So let's do that. And here we go.
How we doing, Bengals? Who wants in? I'm looking around. What is your name, sir? Orlando Brown, is that right?
Yes.
Come on up. Let's hear for Orlando. Great to meet you. Orlando?
Yep.
Got done with the practice. Hot day. Stop the car at a convenience store. You run inside. You close your eyes. I want you to visualize yourself doing this. You open one of the fridges and you grab any drink you want. And you go, nah, I'm going to put back. And then you open a different one and you pick out a different one. And you put it back and you keep changing your mind because you don't know what to do in that moment. And right now, you grab one out and you bring it to the counter. Don't open your eyes. Don't say a word. Folks, think about this. If you were up here, what would you do? Could I have influenced his thinking? Could I have gotten inside his head? I want everyone to see this. Don't open your eyes. Out loud, before you open your eyes, say it. What was the last drink you picked out of there, brought to the counter? What was it?
Orange soda?
Now stop playing. Be for real. What did you pick?
Orange soda.
Are you making that up or are you being for real?
No, I'm being for real.
All right, open your eyes. If I got it every time, it's be a magic show. You know what?
No way. No way.
I got you, Orlando.
Damn. That's orange soda. Damn, dog.
Forget about magic tricks, coach. What if I could look a QB in the eyes and tell you exactly who they're going to throw the ball to next? Now we're talking. Have we ever spoken a word in our life?
No.
All of my wide receivers, tight ends, and running backs, stand up, please. Look at me. Do one look around the room and look back at me, please. Your team, baby. I'm going to show them. Go ahead. Anybody in this room, who are you throwing it to first?
Trenton.
Trenton. Hang back. Throw it back one more time. Now hear me out. One is luck. Double down. Saw that look? Who's next? Go.
Andre.
Toss it. Because I'm already done before you even did. Come on back. Close your eyes. Visualize. Before you get on that field, you visualize over and over what you're going to do. Go ahead. Show me your last play. Go.
Tanner.
Fakes to Jamar. Throws to Tanner. QB, you want to read someone's mind? Let's do it. Wide receivers. Where are you at, wide receivers? Tell everybody, what's your first name?
Tyler.
Stand up. Let's hear it for Tyler Boyd. I want you to close your eyes and picture the face of the first girl you ever kissed. Tyler, any way in the world that Joe Burrow could know this?
No.
I mean, 1% or 0?
0.
I'm going to snap my fingers, and it's going to pop in your head. You're going to hear a voice in your head telling you the name of his first kiss and what grade he was in when it happened. Are you believing this?
No.
No. Me neither. You write down the name and the grade, hold it against your body. When I snap my fingers, I want you to tell him what grade he was in.
5th grade.
No way.
And hold up. Bengals. Was that right, Tyler?
Yes.
If he gets, that might have been luck. If he gets the name, I want pandemonium. I want this place to shake like you just won the Super Bowl. Tell him, what's her name?
Sarah.
Bro, no way.
Show the
No way.
Show the camera.
Bro.
Thank you very much, Bengals!
Okay. I almost forgot to get back. Wait a minute. Hang on, I'm lost here. How do we get out of stop share? Okay, it's amazing what you can learn on ESPN. So again, I'd love somebody to check that out. We'll see if it's real. If it is, I don't think anybody could say that what you think doesn't affect what happens in your life. Certainly seems that way to me. Okay, let's talk about Harold Hillman and what he was telling the world. Okay, so I found these on a website, and the story is that these are the questions that Harold Hillman, who I've talked about a lot, proposed to all the other cell biologists, physiologists, anatomists, all the people who do all the experiments in conventional science and medicine with cells. These were his questions that he had, and he formulated them, I think, throughout his life. And he claimed that when he died, none of these questions were answered ever to his satisfaction. And I think you'll be able to see just how profound this would change the whole practice of cell biology. So let's just go through them. How can one obtain an enriched fraction of a subcellular organelle or cell type? How does one know that the disruptive procedures does not change the biochemistry of the fractions significantly? So here's a thing I got on what is a fractionation of the cells. Biochemical analysis requires disruption of the anatomy of the cell. Gentle fractionation techniques have been devised to separate the various cell components while preserving their individual functions. Just as a tissue can be separated into its living constituent cell types, so the cell can be separated into its functioning organelles and macromolecules, and then they go on. So Hillman questioned this, and he said that anytime you try to get a, quote, enriched or enhanced subcellular fraction, this means that you're going to have to use disruptive techniques like putting in certain concentrations of saline or water and centrifuge and homogenization and all the other things that they do to separate out the parts of this very tiny cell. So his question is, how do you know that these techniques don't change the biochemistry or the anatomy of these individual components? Because the problem is, and he would often say that some things may be unknowable. And because you can't actually do a control, in other words, you can't find them without doing these disruptive techniques. And you can see here that they agree that they're disruptive techniques, obviously. Then how do you know that what you're working with was actually there in real life? And nobody was able to prove to him that that was actually the case. So then you go on to number 3, which is similar. Why does one assume that homogenization and centrifugation, so in other words, putting things in a blender, take the cells, you put them in a blender, and you centrifuge them at a very high rotational speed, do not change the entropy, that's the order, and therefore the free energy and the equilibrium of the reactions in these subcellular particles? How do you know that whatever biochemical pathways or whatever biochemical reactions you get from these subcellular fractions, how do you know that that wasn't at least changed by homogenizing it and putting it through a centrifuge? And as he says, why are not controls always carried out for subcellular fractionation, except for total recoveries relative to crude homogenates? So in other words, they never do these biochemical pathways or look at energy equilibrium with controls so that they would know just how much this homogenization and centrifugation actually changed what you're looking at. Now, here's another interesting one, a little similar topic, but now we're getting into the sort of heart of this. Why is it believed that each biochemical pathway or cycle has its own structural compartment when prokaryotes can carry out virtually all the same reactions in only one compartment? So here I put the definition of a prokaryote as an organism that has a distinct nucleus or other organelles. So these subcellular parts, the parts that are made, that we're saying, we being conventional cell biology says makes up the cell, these prokaryotic organisms like bacteria, they don't have internal membranes, they don't have nuclei that are distinct, and yet they carry out the same biochemical reactions, but they don't say they're in a compartment because they have no compartment. He's asking the question, why do you believe that each individual pathway or cycle, like the Krebs cycle, et cetera, has to have its own structural compartment when we clearly have an example of a organism that doesn't have these compartments and carries out the exact same cycles and pathways? Question number 5, why does the finding of a chemical substance or activity that's located in the same subcellular fraction and a structure identified by electron microscopy mean that the same chemical reaction was located in the particular organelle in the living cell of the intact animal or plant? In other words, if you do all the things that you need to do to find a fraction of a cell, and you take a picture of it in an electron microscope, and you say there's biochemical activity, then comes the conclusion that that biochemical reaction or cycle must have come from that structure. And he's asking the question, how do you know that that assumption is true? And again, I want to point out that if people really are interested in looking into how scientists think and how they get thrown off, I would highly recommend the book on the nuclear atom by Dewey Larson, even though I would say up front that I don't agree with a lot of his conclusions. And I think that even interestingly, he fails to use some of his own investigation techniques in coming to these conclusions. But he points out over and over the fallacies that scientists seem to accept and believe in and run into over and over again. And one of them is that if you have a observation that has more than one possible solution or explanation, as he says, you cannot use that as proof that that explanation is correct. That should be obvious. The example that I've given before is if your husband or wife or significant other comes home late, you therefore conclude that they're having an affair. That, of course, is one possible explanation, but there are many others, like they had a flat tire, or they had to stay late at work, or they went out with a friend, or many other possible explanations. Now, here's the thing. What we hear in medicine and science a lot is, well, that's just one of 6 different observations. And even though none of them actually are proof, because they all obviously have other possible explanations, if you put them together, that the bulk of the evidence or the preponderance of the evidence means that whatever it is has been proven. And I'm here to say that is not the case. It doesn't matter if you have 100 of these things, all of which have different explanations. No matter how many you have, it doesn't end up proving anything. You prove it by proving it. If you want to know whether somebody's having an affair, you have to see them, whatever, having sex in bed or whatever it is, or maybe some other direct proof. You cannot use multiple indirect proofs to prove anything. They do this all the time. And this is one thing, just because a chemical activity or substance is in after you've ground it up and centrifuged it and done all the things that you do to an electron microscope, that does not prove that that process or that chemical was inside that structure, or even that the structure existed in the living organism. Okay, let's move on. How is intracellular movement possible, and why is the viscosity of the cytoplasm so low in the intact cell if there is a cytoskeleton present? In other words, as I've shown you this picture of the cell, and they have the endoplasmic reticulum and this protein network, and the endoplasmic reticulum attached the nuclear membrane to the cell wall. So how can that possibly have nuclear rotation without tangling up the cord? And so, in other words, this movement that you see doesn't make sense based on the actual proteins and structures they say. And where do protein synthesis and hydrolysis occur in cells in which ribosomes and lysosomes cannot be seen? And again, this is something I've gone over. They claim that ribosomes are the site in which proteins are made. It's one of the big principles of the mRNA supposedly turning into protein. They're made in these little factories called ribosomes, which I've shown over and over again are imaginary structures. They're basically gas bubbles on an electron microscope picture. But here he's pointing out that there are cell types that have no ribosomes, and yet they still make proteins. So how is it that you can claim that all protein synthesis happens in the ribosomes? Lysosomes is similar. This is an artifact on an electron microscope. They're said to house the enzymes which break things down, and yet you get breakdown, which he's calling acid hydrolysis, in in cells that don't even have lysosomes in it. So how does that explain? What is the evidence that the microsomal fraction consists of cell membranes and endoplasmic reticulum? So a microsomal fraction, I looked it up, is interesting. Collection of tiny subcellular particles invisible with the light microscope, produced during differential centrifugation of eukaryotic, that's like mammalian cells. Under electron microscope, these microsomes can be seen to consist mainly of membranes and ribosomes from the endoplasmic reticulum. But here it's very interesting. Even these people say the microsomal fraction is thus an artifact produced by the preparation process rather than a biological entity, as interestingly are ribosomes and endoplasmic reticulum. You cannot see them under a light microscope. They're too small. You have to do all the things that you have to do to get an electron microscope picture. And this creates artifacts which they see as microsomes, which they actually admit are artifacts. What they don't realize is that even the ribosomes and the endoplasmic reticulum are also artifacts of the process of homogenization, differential centrifugation, and electron microscopy. And nobody was able to prove to him that that was not the case. This is similar. Why does it assume that homogenization and centrifugation do not affect receptors, their affinities for transmitters, hormones, drugs, ligands, etc.? So all of these receptors, ACE2 receptors, opiate receptors, were all found after doing homogenization and centrifugation of the tissue. And all he's asking for is prove that these very disruptive techniques don't affect how these receptors work. Obviously, they might or they might not. That's something that a scientist would attempt to prove. They never do, and so they were never able to convince him that they don't. So can a particle and a vacuole both be lysosomes? I'm not sure about that one. Can one calibrate substances from tissues you're using pure solutions in simple saline or approximately the same concentration? So if you take a tissue and you put it in saline, how can you calibrate the amount of tissue since it's obviously in a drastically different medium than it would have been in the cell? This one is interesting because we all believe that we've seen cell membranes or membranes inside cells. It's one of the hallmarks of modern biology, and they are studied by electron microscopy, and they are meant to contain lipids. But the problem is when you do an electron microscopy preparation, you must essentially dissolve all the lipids, in which case you would dissolve the membrane. And so he's just asking the question, how can you see the membrane? How can you claim that you're seeing the membrane and proving the membrane exists when you just did a technique to see the membrane, which actually got rid of the exact components that you say are part of the membrane? How is that even possible? And then we go on to more electron microscopy. What is the evidence that this modern rapid deep freezing for electron microscopy causes less shrinkage and distortion of tissue, cells, organelles than classical transmission electron microscopy? And here's a brief description of what transmission electron microscopy is. And this has come up in some of the debates that people will say, well, we know the old pictures of transmission electron microscopy of viruses or organelles, ribosomes, et cetera. They're not so accurate because we had to do all these techniques and staining, et cetera. But now we do the deep freezing, and that doesn't create the distortion. But everybody who knows about if you just deep freeze something, as Hillman often proved, to 160 degrees below 0 Celsius, that doesn't create shrinkage and distortion of the tissue, because obviously it does. Yet they claim that this is a fairly exact representation of what that would look like. So you see all these spaceships and all these other things, and they go through the different components of the membrane or the virus as if that's what it actually looked like. And all he's saying is, prove that to me. And 14 is in a similar vein. We calculate dimensions. So you'll see, viruses are this size, or cell membranes are this size, or the synapses are this size. And we get that from the electron microscope pictures. Yet we don't account for the shrinkage during the preparation and examination of these images. And so, and one of the reasons they do that, he says, is because they actually don't know how much it shrinks. And so rather than saying, we just can't use this to make any sort of measurements or calculations, that would be what an honest scientist would do. They just pretend like it didn't shrink, and so you get an accurate measurement. And it's true in some cases, probably in all cases, you don't know how much it shrank, but then you don't go and assume that it didn't shrink, and so you get an accurate measurement. But that's what happens in modern cell biology. And then, do membranes in cells appear to be normal to the plane of section more often than solid geometry would permit? And when you read Hillman, you get into this a lot, where it seems like pretty much every picture, electron microscope picture of cell membrane, is a perfect line that he says is normal to the plane of the section. Yet, if you realize that you were cutting through a three-dimensional object, you could never get all the pictures to be in the same plane of the section. That's simply not possible. In other words, if you have an orange and you homogenize it and slice it randomly, or even with a microtome, so you're slicing it so-called regularly, you would never have all the pictures of the components line up exactly normal to the plane of the section. But that's what you see on most of the pictures of electron microscopy. You don't see them going in all different directions. And so that should raise red flags that you're not seeing something that was there in real life. And so what you're seeing is, of course, an artifact. And okay, then you get into 16. Can you know the thickness in life of any biological membrane? He would say no. Nobody has proved that we know the thickness, because in order to get an image of it so that you can measure, you've had to disrupt it and actually dissolve the very component, that is the lipids, that you say it's made of. So how can you possibly know the thickness? Why should it be necessary to tilt the stage of the electron microscope to see randomly oriented membranes in all orientations when it's not necessary with a light microscope? So when you do look at membranes in a light microscope, they look all different arrangements. They seem to be randomly oriented, as you would expect if you homogenized, in other words, put an orange into a blender. But in an electron microscope, apparently you have to actually tilt the stage, which is not the sample, in order to see these random orientations. And that, of course, makes no sense. 18, we'll keep going. How can carriers assist the passage of ions, amino acids, etc., across membranes when the combination must be bigger than the substance carried? Let me just read the next few. Why have few or no carriers been isolated? What is transport? Why are receptors and channels which have been characterized, sequenced, and their size measured or calculated not seen on membranes by transmission electron microscopy? So all these have to do with this question that I've been asking, and which obviously has come up recently with COVID, and it's a big deal for opiate receptors. It's a big deal for ACE2 receptors. It's a big deal for sodium potassium pumps. He's saying, first of all, there are none of these carriers have actually been isolated. And second of all, you say they're a size which you should be able to see with any kind of electron microscopy, including transmission. Why don't you see them? Why are all the pictures that I've showed people over and over again of these ion channels and these membrane-bound pumps and membrane-bound receptors, why are they all, schematic drawings and computer images and CGIs and all these sort of make-believe, people make up what they look like, or they do X-ray diffusion, so they get this vague image of some X-ray shooting into a membrane, which they haven't isolated it, so they don't actually know what they're shooting into, and they get this scatter form, and then they make a pretty picture out of it, and they call that the membrane-bound pump or the carrier or the ion channel. We see these even in, the calcium-gated voltage channels, which are supposedly such a big deal with EMF toxicity. We've heard a lot about those. And just to remind people, I'm not saying that EMFs are not toxic. I'm talking about the mechanism that it doesn't work on these calcium gated ion channels because they've never been identified, isolated, photographed, or even though they're the size that you should be able to see them with electron microscopy, there are no actual photographs showing these carriers, these pumps, these channels in the membrane. So why is that? He's asking, show me something that I can prove to me that it exists. Now, again, that doesn't mean that EMFs don't cause harm. It doesn't even mean that, for instance, there may not be changes in the calcium levels in or outside of the cell or tissue. It doesn't mean that. We're talking about mechanism here, and we're talking about the claim that this is based on membrane-bound receptors or channels. And you could say, so what's the difference does it make? You know, it causes trouble. So isn't that good enough? And I would say that's not good enough. We're supposedly doing science here. We're supposedly figuring out mechanisms. And I would be very happy with forgetting about this whole business of, you know, this sort of subcellular microscopic transmission EM based biology, but that's actually what's driving the whole biomedical complex. And I keep saying over and over again, the only biology that makes sense is to actually study living organisms with various influences on them, with minimally disruptive or no disruptive techniques, and actually getting your information through that way, which will be the best and most reliable information. So this way of thinking is actually a call for a whole different way of studying living things, a whole different sort of consciousness about what that means. And it will, by necessity, get us into a more living biology and the questions of what things in our environment affect actual living beings. All right. 22, can an electron microscopist looking at a metal deposit on a biological structure derive any information about its chemistry? So in other words, when you see an electron microscope pictures, not the deep freezing ones, but the usual ones where they're using metallic stain or osmium stain, they're just looking at metal deposits. And they're claiming that these are structures and that these structures contain within them a certain chemistry, but obviously even adding a metal to a structure, even if the structure isn't even there, or even if it is there, would obviously change the chemistry. And so how do you know that it didn't change the chemistry if you put a metal deposit on a structure, which is what they do. And then they study that and they claim that it had no difference, had no impact on the chemistry. And he's actually just asking them to prove that. Now, keep going. Why do lamella of the myelin sheath appear to be equal distance apart, irrespective of the thickness or depth of the longitudinal section cut? So here we're looking at a schematic diagram of a nerve, and it has this fatty coating called a myelin sheath around it. And here you can see it in another orientation. And he's saying that when they show you these pictures, they all are exactly equal. Like here, if you put this out, each one would be equal. And he's just saying, if you have a nerve and you cut it randomly, because obviously you can't cut it perfectly longitudinal to the nerve, you're never going to get all the sections to be exactly the same size. It's just like if you cut a rope and you cut it randomly and you cut it, it's going to be somewhat diagonal. And so the sections are never going to be equal distance. Yet that's what you see. And so whenever he encounters a difference between the geometry that you would expect and the geometry that you see on an electron microscope picture, that should bring up red flags that you're not actually seeing the thing you think you're seeing. You're seeing an artifact which probably would be regular because it may be just a freeze fracture or something, which is much more likely to be oriented in a normal way. In other words, longitudinal, because that's just how it would freeze, and that each section would be equal, but that can't represent an actual living structure created through homogenization. And then he just asks, is this repeating distance sufficient to regard it as a good model for a cell membrane, which apparently they do. Then he says there's often a difference between what you see on a light microscope, which he tends to believe more. So here the myelin sheath is darker than the cytoplasm, but in an electron microscope picture, it's not darker. So why is that? And you often see these discrepancies, like you'll see the things that can be seen in a light microscope turn out to be completely different size than the size you see in an electron microscope. Now, this could be for a lot of reasons, including shrinkage, whatever, or expansion, or just the way that it's processed. But they often will measure it, the electron microscope picture, again, as if it's the truth, even though they get a completely different number measurement from a light microscope picture. And he just is pointing out that one of those has got to be wrong. And why do we assume that the one or the other is correct? And here we keep going with the why is it assumed that receptors are on the surface of the cell membrane? How valid is the use of agonists, antagonists, ligands to detect receptors instead of the transmitters, hormones, drugs, toxins themselves? In other words, with many of these studies that look at these receptors and ion channels and pumps, they can't be studied with the actual hormone or drug that supposedly binds to them or goes through them. They have to use different antagonists or agonists. That means it's the opposite of antagonists, or different chemicals called ligands to detect them, not the actual thing. So why is that? And why is it assumed that they're on the cell membrane, these things, or embedded in the cell membrane, these receptors or channels, when they're not seen in the cell membrane? Why can't they just be a binding principle of the cytoplasm? And in fact, there we do get into the principle of there is another possible explanation. Therefore, it can't be used as the proof that there are these membrane-bound receptors. Moving on. Why are the dimensions and numbers of synapses different by light and electron microscopy? So here's the same thing. So you see these pictures of gaps in nerves, but the dimensions are radically different by light and electron microscopy. The numbers are different, which should make you think automatically that one is not an accurate or realistic picture of what is actually there in life, or maybe neither of them are. So what would make you believe one and not the other? And both of them involve dehydration and therefore a break in the nerve axon, when maybe that's the reason you get these synapses, these gaps, and you do more of this dehydration and essentially manipulation of the tissue with electron microscopy. Therefore, you get different size gaps. Isn't that a more likely explanation than somehow you're seeing different size synapses or gaps depending on how you look? Why are there no light micrograph pictures in the literature? And I'll have to take Hillman's word for it, but he was incredibly meticulous, showing the connection of one cell body by a dendritic presynaptic fiber to a synapse on another cell body. And in many of my talks, I've talked about, I looked through the literature to see one light microscope picture of a nerve connecting to another nerve, the cell body to the axon by a synapse. This is one of the foundations of modern neurology and neuroanatomy. Was not able to find them. So apparently he couldn't find them either. And he's just asking, since you say they're big enough to see, according to the measurements that you're getting with electron microscopy, why don't we see them under light microscopy? And that's often the case. You'll see they'll see a structure on electron microscopy. They measure it, claim that's the actual size of it. That size would be big enough to see with a light microscope, and yet there's no pictures of it with a light microscope. So he's simply asking the question, why is that? Why don't we see it? You say it's this size, we can see things of that size, we can prove that. So why don't we see it? And the best answer for that is because it's an artifact of the electron microscopy process. And again, if you're sitting there wondering, like, this is pretty wonky, esoteric, useless, irrelevant stuff, this is the foundation of how a neurologist thinks about how a nerve works and how to treat you, based on how your synapses are working and how your myelin sheaths are functioning. And so if you don't think that having a completely wrong conception of whether these things actually exist and how they function might not interfere with the end result in the treatment, I don't know, doesn't make any sense to me. Then he asks the question, does the chemical theory of synaptic transmission contain unprovable and unproven hypotheses? It's a sort of a weird question, but if there's no synapses and the whole theory of chemicals traversing the synaptic junction, that's the whole rationale for SSRIs. One of the most commonly used, 40 or so million people take these drugs based on this theory. And he's essentially saying, we can't even see these synapses. We can't measure them. We think they're artifacts if you really look at them. And there's all kind of hypothesis about how this works that to his eyes were unproven or unprovable. And so maybe we need a different model. Question 31, why does it assume that evidence derived from experiments on neuromuscular junctions is relevant to transmission in the central nervous system? So that's just a case of doing experiments on the connection between nerves and muscle, and then saying that must be the same mechanism and anatomy that happens in the central nervous system. And that's not necessarily true. This question 32, I've also talked about a bunch of nuclear pores allow RNA to pass through. How do they prevent smaller molecules and ions going through at the same time? Why is there a potential difference across the nuclear membrane? And this goes, if you look at the conception of the cell, it has a nucleus in it, and then it has a cytoplasm. And these are things you can see under a light microscope and in live blood cell. And if you put a pH probe in the nucleus and in the cytoplasm, they're different, which means that the hydrogen ions, which is the quote smallest quote thing that we know of, cannot freely diffuse from one compartment to the next. If they could, the pH would be the same. So an RNA, an mRNA in particular, is thousands or maybe millions of times bigger than an hydrogen ion. So how does it get out of the nucleus, which is supposedly the mRNA is made in the nucleus and gets out of the nucleus and goes to the imaginary ribosomes? How does it do that when the whole doesn't even let something that's 1,000 times smaller get in and out? This is the same thing that I've talked about over and over again. How do you build a door for elephants that keeps the mosquitoes out? The answer is you don't, and there are no nuclear pores, and the RNA is not made in the nucleus and goes to the cytoplasm. There are no ribosomes, and there's some probably water-based thickening of the nuclear, essentially the nucleus has a different crystalline water structure than the cytoplasmic water. So there's like two different crystals, and as a result of that, the hydrogen ions stay in one compartment more than the other. There is no membrane, there is no channel, there is no pump that keeps them out. That would be a grossly inefficient energy expenditure way of doing it. And the body doesn't need to do that. All it does is have to make different crystalline compartments out of the water and the protein. And that's enough to keep things out that it doesn't want and keep things relegated to their proper place. Okay, now we get into some questions. What is the evidence that each cell of a particular plant contain the same quantity of DNA? In fact, there is no evidence. They have different quantities, different types. There is no central genetic code that determines life. There is no same DNA in all the different cells and tissues of our body. This is, I would say, an experimentally proven fact. And as Larson points out, one of the problems with all the sciences is they come to conclusions which then become sacrosanct. And then sometime later, we get more experimental evidence that these conclusions are incorrect. In other words, they conclude that the DNA is the same in every cell of the body. And then they did that at a time where they couldn't actually evaluate the particular DNA in each different cell. And now they have some ways of doing that, although that's problematic too. But even in the problematic ways, they clearly get different DNA in different cells, but they don't go back and change the original conclusion. They do in their own circles, interestingly, and then write papers about, well, someday we have to refine this. But in all the things that go out to the public, all the things in the textbook, all the things that are taught in high school and college, they just go on pretending as if the old conclusion was still valid, even though they actually know it isn't. If the cell membrane is fluid mechanically, how can cells maintain their integrity? So they say they have these fluid membranes, but yet the cells maintain their integrities. He's just asking how this is possible. In immunocytochemistry, is it assumed that the fixative, dehydrating reagents, washings, primary and secondary antibodies do not change the reaction of the antibody to the antigen believed to be in a particular cell or part of the cell? It's again the same question. A lot of these proteins, like the spike protein, have only been found through the use of antibodies. And he's asking the question, doesn't the things that we do in order to prepare the tissue or the cell or the protein soup, all these things, don't they change the binding capacity and make it essentially a kind of irrelevant experiment, or at least one that you would have to control every step of the way in order to come to a conclusion? And as I pointed out, particularly with the spike protein, they don't do it with different antibodies. They don't do it with things that don't have the spike protein and use the same antibodies. So it's a very anti-scientific process, and he's just pointing that out. 36, I can skip. Under what conditions can tissue cultures be used in the study of tissue from which they originated? And here he's alluding to this, one of the central questions, which we've also talked a lot about. We get these tissue cultures, and that's the only way they can, quote, isolate a virus. And he's just saying, under what conditions do you say that these tissue culture actually gives an accurate and fair representation of the original tissue that it came from, seeing as how many of the tissue cultures have been around for 50 years and are highly inbred and they're removed from their living matrix, and on and on and on. So what makes you confidently assert that it will have the same reaction? They do these with cancer studies. They study tissue cultures, and they claim that it will have the same reaction as that tissue in a living person. And he's just asking them to prove that. And again, is it warrantable to assume that growing tissue and culture does not change how they react, their biochemistry, what they look like, or their immune system, immunoreactivity, not their immune system. But we assume that it doesn't. And so you get all these studies that people show you on the immune basis of different tissues based on these tissue culture experiments without asking this fundamental question: did the tissue culture change the reactivity fundamentally from what it was in real life? These are some questions. Then it goes into the different types of nerve cells, which that was his field, neuroanatomy. So I won't go into those, but they're sort of interesting to look at. And he's looking at question 41s. These cell types, which he says don't exist. There's very few cell types in the nervous system, and I don't remember which ones he says are real, but not all these different astrocytes and oligodendrocytes and microglia. Those are different antibody staining techniques, which he says produce artifacts. And you get into the question 42, there's no agreement about staining procedures which are used to identify these. How come we can't agree on which stain produces which one? And so that's also a red flag. Why is it necessary to use tissue cultures of alleged cell types to identify them and their markers? Why can't you use the tissue directly? And that gets into the problem I just said. Doesn't a culturing of a tissue and supposedly growing it completely out of its living matrix essentially change everything? And so how can you rely on that to give you information about markers and how it's functioning? Here he gets into 44, questioning whether all cells have the same DNA, because some make different proteins. Then they say it's because of suppressor genes. And he's just saying, is that the only possible explanation? Maybe there's different DNA in different tissues, or maybe it's not the DNA that makes the protein. Again, we get into the problem of anytime you claim that something proves something, but there's other possible explanations, that can't possibly be the proof. And finally, if diseases are believed to be autoimmune, either organ or tissue specific, why does the body not reject the specific organ or tissue as it rejects incompatible transplanted hearts or blood in the wrong group, often making the patient ill or even killing them? So this is a question: if there is autoimmune disease and the basic model is tissue rejection when transplanted with an incompatible donor, in that case, it basically completely destroys the tissue. But in autoimmune disease, you just get an inflammation. And he's just asking, what is the basically mechanism for this difference? Where is the explanation of this? Why are pure proteins used for calibration when different tissues contain different mixtures which have different calibration curves? This gets into the question of identifying specific proteins and what they actually do. And they're all taken out of their milieu and they're calibrated when they're exactly pure, or as pure as they can get them, which is never the case in real life. They're always mixtures with different mediums, living matrix. etc. So we're again getting an artificial calibration. And then finally, why do synapses seen on electron microscopy appear so much smaller than those by light microscopy? Again, you see different sizes of things depending on how you look. And obviously that has to do with the technique. And he's just asking, how do you know which is the right size or whether that structure actually exists? And doesn't that make you wonder, if you keep getting different sizes of things, that maybe there's something wrong with this picture? So that was a lot. And I hope that basically the idea you got was there's some fundamental questions which Hillman was a master at coming up with, which really talk about the fundamental problems of the way we do biology and study living things. And as I've said, my hope is that we study living things in their natural habitat with as few, if any, disruptive techniques. And our goal, whenever we study a living thing, is to somehow enhance the life of that thing. Otherwise, I'm not sure we should be studying it. So I hope everybody at least got through this or enjoyed it a little bit. And I will see you next week.